Okay, hi guys. So this is a sample of a question that you will get, which is an experimental technique question. So what I want you to do is pause the video and try the question. And when you're ready, try the question and answer it. Okay? All right, so let's look at this. Okay, you ready? So in this question, they prepared two test tubes, okay, A and B, and both of them contain water. Okay. They also have a partially permeable membrane. A partially permeable membrane is the same like your cell membrane that allows only small, simple molecules to pass by. Whisking tube A, this tube is called a whisking tube. It only contains starch. Whisking tube B contains starch plus saliva. So saliva usually contains amylase. So amylase will break down the starch, right? So the apparatus was left for 24 hours. After that, samples of water from each test tube were taken. Describe, giving practical details, how you will carry out a test for starch and a test for reducing sugar. So starch, you will use iodine solution. So iodine solution by itself is a brown or yellow solution. So a positive result means the yellow solution turns dark blue. Okay, reducing sugar. Reducing sugar means to test for glucose. So we use Benedict solution. You add Benedict solution and then you heat it up. So the Benedict solution itself is blue. All right, if there is sugar, the color will change to red. All right, so if it's red, it means that there is sugar. If it's not red, it means there is no sugar. So over here, remember starch is big and nothing happens to the starch, so the starch can't go out. So inside the solution, you will still have starch. So in test tube A, in the starch suspension inside the whisking tube, yes, you will have starch. You won't have any reducing sugar. In the water, do you think you will have any starch? No, because starch is too big to diffuse out of the starch is too big. Remember, starch is a big molecule, it cannot go out. And there is no reducing sugar because there's nothing to digest the starch. So nothing happens. There is only your original starch. For test tube B, saliva will break down the starch to form glucose. So what happens is, after 24 hours, will there be any more starch? So no, there will be no more starch. Okay. The sugar, some of the sugar will go out because the sugar is small, glucose is small. Some of the sugar will stay inside. Okay, because not everything can go out. It will only diffuse until it's even. So inside the water, you will have reducing sugar. We will have reducing sugar here means glucose. You will have glucose, and in the whisking tube, you will also have glucose. But in the water, will you have any starch? No, because starch is too big to go anywhere. And there is also no more starch. All the starch has been digested. Okay? Explain your predictions for whisking tube in B. So why is there no more starch? Okay, so the starch is all broken down by amylase in saliva to form glucose. Okay, so to get your marks, starch is broken down by amylase. Okay, and then some amylase remains in the tube, remains inside. because you can't have all the amylase diffusing out. Some of it only can go out. All right. Okay. Why is it the water in test tube B will have glucose? Because glucose or sugar molecules are small enough to diffuse into the water. Starch molecules are too big. Okay, so read through the question again and read through the answers again. So these are the questions when we talk about, that you will see when we talk about experimental questions. Okay, so I will see you soon. Bye-bye.